So our next speaker is Brianna Johnson. Brianna is a second-year master's student in the Wildlife Resources Department at Utah State University, and she'll be talking about symptom progression and serological dynamics following introduction of a low-virulence Mycoplasma ovipneumoniae strain in a desert bighorn herd. Brianna, are you able to load yourself up?
I think so. Thank you.
Great.
Can you hear me and see my screen?
Yes, we can. Please go ahead.
Wonderful. Okay, so thanks for that intro. I'm Brianna. I'm a master's student with Kezia Manlev at Utah State University. This is my first Northern Wild Sheep and Goat meeting, so I'm glad we were able to make it happen this year, even if I didn't get to go to Canmore. I'll be talking today about the specific details of a Mycoplasma ovipneumoniae disease event in a desert bighorn system. Of course, we all know that respiratory disease is of enormous conservation concern for wild sheep in North America, and Mycoplasma ovipneumoniae is the primary causative agent of this respiratory disease. When it's transmitted to naive wild sheep, it contributes to polymicrobial pneumonia and typically elicits an immune response that is quite detrimental to the host. These disease events are often severe and population limiting, and we typically expect to see years of reduced recruitment as outbreaks occur in new lamb cohorts. I'm working with the desert bighorn herd in and around Zion National Park here in Southwest Utah. The current bighorn range encompasses the southeast quadrant of the national park and extends to the south and east in continuous cliff and canyon habitat. Bighorn sheep are native to the Zion area. The original herd was extirpated by the 1950s, likely due to respiratory disease. A handful of animals were translocated from southern Nevada in the 1970s, and the herd has grown pretty steadily since the translocation. The most recent population estimate was almost 900 animals. Individuals close to the center of the herd were observed with respiratory symptoms in July of 2018. And those individuals subsequently tested positive for MOV. Intensive monitoring of MOV disease progression has not really happened in a desert system before, and Zion is a good focal system for this kind of disease event because of the high frequency and reliability of observations. There's pretty good access to the bighorn habitat, and perhaps most importantly, because of the high visibility of this herd and the regular attention they get, we're able to pinpoint pretty closely when the pathogen arrived, and there's been reliable monitoring from the start. As I talk about the details of this disease event and why it's unique, there's a few things I'd like you to keep in mind. First, we know that MOV arrived at Zion in the summer of 2018. We know that some animals are showing symptoms. We know the pathogen is present and circulating. We do find it every time we look for it. And we know that animals are producing antibodies to the pathogen. We have looked for other organisms and immune signals in this herd, but not found a pattern that aligns well with the symptoms that we're seeing. Here's a quick sample of some of the routine testing that happens in this herd. So there are three primary ways that we're tracking this pathogen in the Zion bighorn herd. We look for symptoms of respiratory disease, we look for the pathogen itself, and we look for evidence of the pathogen in the form of antibodies. As I talk about the monitoring a little bit more, I'll show examples of what has been reported in some of the other systems we've heard about today, and use those to contrast what is happening at Zion. A number of symptoms are relevant and together used to develop a symptom score for each observation. It's an example of a lamb coughing. This is what the symptom scores look like. The Black Butte herd in Eastern Washington has experienced years of disease and reduced recruitment. These observations are from 2013 and represent pretty well what's expected in lambs born to infected herds. Here we see a pulse of symptoms beginning in early June, continuing for a few weeks, and then the observations end when all the lambs die by the end of July. This is the same scoring system applied to the 2019 lamb cohort at Zion. These were the first lambs born after MOV invasion. we can see symptoms again emerging in June, continuing through the summer and sort of tapering off into the fall. And these lambs don't experience the mortality seen in Black Butte. The initial timing and structure of these outbreaks looks sort of similar, but these lambs were not born at the same time of year. Mean parturition at Zion is early March, while mean parturition in Black Butte is mid-May. So if I adjust the time axis to a line parturition, we can see that in 2019, the Zion lambs are symptomatic not only later in the calendar year, but substantially later in their lives. And in 2020, lambs at Zion showed almost no symptoms, even though the number of observations actually increased. This pattern of a year of mild symptoms following a year of severe symptoms has occurred in other places. So it'll be interesting to see what happens at Zion next year. Moving on from symptoms, current infection is detected with a PCR test performed primarily on nasal swabs. These results are reported in cycle threshold, which is the number of replications required in the PCR assay to amplify pathogen RNA to a detectable level. The important takeaway here is the inverse relationship between cycle threshold and pathogen load. So a high cycle threshold means there were fewer gene copies in the original sample, and a low cycle threshold means the target is more readily detected. And with that inverse relationship in mind, I'm going to use cycle threshold on PCR tests as sort of a proxy for pathogen load. This data set from the Snowstorm Mountains in northern Nevada is a good example of cycle threshold increasing through time as a herd moves towards clearing a pathogen. So you can see the pathogen load pretty clearly decreasing through time. Another example here is from Hardware Ranch, which was the captive population in northern Utah earlier this year that Kezia talked a little bit about. And though the observation period here is brief, the Hardware Ranch sheep also show a rapid trend of pathogen clearance. And this is what the cycle threshold from Zion looks like. So really gentle downward slope here that looks more like maintenance than clearance. We also see lower MOB prevalence at Zion compared to these other herds. Over these same testing periods, 53% of PCR tests from Hardware Ranch came back positive, 61% from the snowstorms, and only 19% were positive out of Zion. When we look for evidence of exposure to the pathogen, we use a C-ELISA test. The serological test measures antibody response to MOV measured in percent inhibition. And this is a positive relationship. A higher percent inhibition is generally taken to indicate a more robust immune response. Back to the Snowstorm Mountains. This chronically infected herd consistently produces pretty high percent inhibition values. Here at Hardware Ranch, we see a dramatic increase in antibody response immediately after pathogen introduction. Again, even over the brief time period that they were captive. And here's what we see at Zion. I think the important thing to note here is that the antibody response in the snowstorms is consistently high, quite a bit higher than Zion has gotten. And that the antibody response at Hardware Ranch quickly surpasses the response seen in Zion. The antibody response at Zion has been increasing, but it hasn't happened as rapidly as in the Hardware Ranch sheep and hasn't reached those high levels seen in the Snowstorm Mountains. All of our diagnostic testing is performed at WADDL. As Kezia described earlier, the serological test results are usually reported categorically. A sample is considered to have detected antibody when the percent inhibition is above 50%. A sample is considered indeterminate when percent inhibition is measured between 40 and 50%. And a sample with a percent inhibition measured below 40% is placed into this not detected category. Anybody who's submitted samples to WADDL for testing has probably seen a report that looks a lot like this. This is just a reminder that even though we're used to thinking about these results in a discrete way, there's continuous information here in the form of percent inhibition. Here is another way to look at MOV serology at Zion through time. Here we see the antibody is pretty clearly not present before 2018. Values are rising in 2018 and 19, but as of 2019, most individuals in this herd would not be considered infected if we're following those interpretation guidelines. Even though we know the pathogen is present, we do detect it by PCR, and we do measure some antibodies in animals are symptomatic. These interpretation guidelines also suggest that a bighorn herd that has been exposed to MOV will have more than 30% of animals with an antibody response that is measured in that detected category. This is what the categorical test results look like through time at Zion. Animals are producing antibody response that places them in the indeterminate category in 2018, and a few make it into the detected category in 2019. But in the 2 sampling years after a known pathogen introduction, the Zion herd still doesn't have that 30% of animals in the detected threshold like we might expect. But again, some animals are symptomatic and we know the pathogen is circulating. So back to these things I asked you to keep in mind. Each of these certainties needs to be conditioned. Symptoms are present, but emergence looks different from other disease events, and symptoms are occurring less frequently. So far, the pathogen load has not been decreasing as expected, especially compared to Hardware Ranch and Snowstorm Mountains herds. Animals are producing antibodies, and production is rising, but less dramatically than in other disease events. And even years after exposure, Zion does not meet the current criteria to be considered a positive herd. Basically, this disease event looks different in every way we've measured. There are a few things we think could be contributing to this. There are many factors that can contribute to the way a disease presents in a population, and there are a few of these that I think are particularly relevant to what we're seeing at Zion. First, we've seen there's a lot of diversity within the pathogen itself. The strain we have here is in this group with strains that are thought to be less virulent. We do see substantial differences in things like birth pulse timing and duration, and in group composition between desert and Rocky Mountain subspecies. And as I mentioned earlier, most of this kind of intensive monitoring has occurred in Rocky Mountain bighorn herds. Essentially, we know that some combination of factors is impacting the way bighorn sheep at Zion are responding to this pathogen. It seems possible that combined host ecology and low pathogen virulence are resulting here in a strategy of tolerance rather than resistance. In a resistance strategy, we would expect to see a robust immune response that is costly to the host. And of course, the goal of this resistance is to effectively clear a pathogen and return to a healthy state. But with a tolerance strategy, the host allows a pathogen to persist, and we don't see that costly immune response. What we might expect to see if this were happening is relatively stable host condition and low mortality, along with symptom, pathogen load, and serological dynamics that look a lot like the patterns I've described at Zion. My goal here is not to totally disrupt the MOV disease paradigm, but to update and refine our understanding of how this disease operates under this particular set of circumstances. Obviously, this could have interesting management implications. So of course, I think this warrants further research specifically into low virulence MOV disease events in a variety of systems. I've worked with and received support from the National Park Service, the Utah Division of Wildlife Resources, and the Zion nonprofit partner. I've borrowed some data for this presentation, so I have lots of people to acknowledge. And with that, thanks for your attention. I'd be happy to take any questions here if we have time, or if you prefer, you can send me an email.
Great. Thank you very much, Brianna, for a nice presentation. We do have about 5 minutes here for questions. So if there are any questions, please, we'll take a minute and allow you to get your questions in the chat line. And sorry, not in the chat, in the Q&A, please. Yeah, and while we wait for that, Brianna, did you, I don't think you said anything about genetic diversity in this herd other than a broader comment about genetics. What do we know about that? And is this playing a role?
So the transplants at Zion are from the River Mountains in Nevada, which is a source for a lot of desert translocations. Right now, I'm thinking more about differences that might just come out of subspecies.
Right, right. And any behavioral differences that may be influencing this?
Yeah, the sexual segregation seasonally seems to be different. Group size can be different, and there's that major difference in the birth pulse. We don't see nursery groups that are quite as big as in Rocky Mountain herds, and the difference in age between lambs can be really dramatic. So I think those dynamics are probably quite a bit different.
Interesting, neat, great. All right, I think I don't see any questions coming in right now, but there may be some coming, so you'll remain as a panelist and people will be able to ask you questions.
I'll keep an eye on the Q&A.
Yeah, and thank you for catching us up here.
Thanks, everyone.
Thanks. All right.